then let's start getting to work. So the first thing to do as an exercise is to log on to the HPC. Everyone who registered previously should have gotten an email last Thursday, I believe, telling you to make sure that you could log into the HPC without problems and that you have the right tools to do so. Go ahead and please log in using whatever tool is appropriate. If you need to follow the link below, feel free. If you have problems, put up a red sticky and we will come help you. Once you're logged in, I will want you to follow the directions below. Or sorry, not the directions below, this is the output. These. Once you're logged in, you can just either copy or paste that or type them individually, it's up to you. And I'll wait for green stickies and red stickies. Okay, so we're mostly green. There are a couple of exceptions, but I'm going to go ahead and start talking anyway. And we're going to move on, and if we get too far behind, just throw up your hands that I know. I probably shouldn't be quite so tongue-in-cheek, but for those of you that are not HPC users, I'm going to be showing you a lot of things that if you're not used to it, it's going to look like gobbledygook and gibberish, and you probably shouldn't worry about it too much. The way I teach this class is to make sure that you have a series of commands that you can run, but I am not trying to teach you what the commands mean or why. That is the point of, say, an introduction to Bash or Linux class, which we also teach if you want to take one later to understand what we did so that you can be more stable later. But all I'm trying to do is give you the commands to run to actually do this thing. After we get to that point, you can try to learn and we will do what we can to help. I found that this is much more effective because it gives you a thing that you can do. So for those of you with green stickies up, do you all have a connection? Has the login, the .login part finished and you have an actual ability to type afterwards? It doesn't say waiting still, something has come back after that? Okay. So those of you that logged in, you should be able to do these two commands. Once you've logged in, it sets you back to your home directory. We're going to change into the RNA-seq model directory and go to ls-l. ls-l just shows you what files are in that directory. This is actually out of date. There's a bunch more things in there now. But you should see something that looks more or less like this. You'll notice that there is a directory called data. So if you cd into the data directory, you will see a bunch of things. So I'm going to try to do this myself now. So what you see, or what you should see, is what I see, which is that there are 4 sets of samples here. We've got everything, well, 2 sets here that start with the R's, and with 25 and 26, and then 82 and 24. These are our example conditions. So these are previously published RNA-seq studies in a human reference looking at difference in gene expression between salivary glands and lungs. So if you haven't spent a lot of time looking at RNA sequencing data, the most important bit here is that what you have is a FASTQ file. FASTQ files are incredibly frustrating to those of us that think about data a lot and think about sizes of things. It is 4 lines long for every individual read. So we have the name of the read, which is a long, incomprehensible string of characters, the actual read itself, a line that is mostly blank, but sometimes will be a copy of this. It's called the notes field, and nobody uses it for anything. And even when they do, nobody pays any attention to it because it's a notes field for a record that is several million records long. Why on earth you would need notes about individual reads? Nobody knows, but there it is. And then this last line is the important line, at least the first part of the most important bit, which is the quality line. So every base that is read, that is generated by a FASTQ or by an Illumina machine, generates a base and a quality associated with that base. We're going to take advantage of that next. So we're all ready to go. We can go ahead and start running our RNA-seq, except no, what did I say? Quality assessment, quality control. Make sure your data are good before you do anything else. There are a whole bunch of different ways that you can go about this, and it kind of depends on how much you want to check on your data before you do anything. The quick and dirty two pathways here, either you can do Trimmomatic first. Trimmomatic is your quality control step. Trimmomatic is a tool that will get rid of adapter sequences, trim your reads based on quality metrics, and output your data back to you in a usable format so that you can actually continue on. FastQC is a quality control or a quality assessment tool, really. People do things weird in names. It is a tool to take your reads, look through them, and tell you what the quality of that data is so that you can actually move forward. So either if you believe that your data are decent and you just know that you want to get rid of junk and move forward, you can do Trimmomatic first and then FastQC, or you can do FastQC and then Trimmomatic and then FastQC again. The second option here is just let's look at our data before we do anything to it, and then then do something to it, and look at the data again and decide whether or not we like it. So here's what we can do. So we're going to go ahead and do this exercise pretty quickly. I'm going to describe what it is, but feel free to copy and paste this set of commands while I talk about it. So I mentioned that every FASTQ record is 4 lines long. The head command that we're using here prints the first n lines of a file. So head-n, we are going to look for printing 400,000 lines out of these 2 files. For those of you who are math inclined, how many reads are we taking? If we have 4 lines per read, how many lines are we taking? Divide that by 4. So we're taking, 4 million. So we're taking 1 million. No, I did it right the first time. 100,000 reads of this first entry. When you go home to your own data and start doing this, don't do this. I'm making you do this so that we can run some experiments and do some work live. The amount of time it takes to actually trim a line, do all of the top hat and the read counting for a full 50 million reads is going to be probably a couple of hours per sample. And if we just hit return and then waited for that to come back, we would start again sometime around 5 p.m. tonight. So instead, we're going to subsample. We're going to take a little bit of the data to show how we could do the work, and then later, overnight maybe, we can get the full sample to run, and you can see what the actual results would look like. So in the meantime, we're going to take this first sample name, which is the 15325, and we're going to take those subsets. For bonus points, anybody know what the _1 and _2 refer to? Yes, the forward and reverse. So make sure that you're always checking on these. Yet another, by the way, pet peeve of mine is the fact that these are in separate files and that the names are complicated and difficult to follow on, and that all of the tools require that you put both file names in. There's no reason for that. Nobody's going to name their forward file something and then their reverse file something completely different. If you were thinking about doing it, don't. It doesn't make any sense, right? So we're going to just take those, and because it is the first sample, and I downloaded it and I know what it is, this is a salivary gland replicate. So of the conditions that we're talking about, there was salivary and lung. This is the first replicate of the salivary experiment. So we're going to name it appropriately. When we take a pause, now while you guys are doing this, and please feel free to put up green stickies while I talk once this is finished, Why are we renaming it to salivary underscore anything? If you have samples, they're going to come back from your sequencing center with some sort of random string of characters that have to do with what lane it was, what run number it was, and maybe what multiplex it was on that lane. That is completely uninformative. At a bare minimum, and there's a whole other range of things that people can talk about when you talk about data carpentry, at a bare minimum, you need to, as soon as you get your sample somewhere, make yourself a file in that same directory that tell you what file name correlates to what sample input you had, right? As a data tracking device, as a reproducible research device, you need to at least know what your sample name means in terms of what your conditions are. Once you get past that point, once you have your data, the next thing to do is to make sure that the names make sense. The frustrating part here is that as you do more and more work to the same samples, you're going to have to add more and more descriptors to your file name. We'll see a little bit of that as we go through this. If anyone here has played with tools like Mother, it is the queen, I guess, of doing that, in that you will end up with file names separated by periods that stretch out through every test or every function that you applied to them to make it 15 or 20 words long, which is really frustrating to deal with when you're trying to deal with the data. On the other hand, when you come back in a year and haven't touched this in a year, at least if you remember how the tool works, you'll be able to go back and look at what happened without having to start from scratch. Almost everything I'm going to tell you about reproducible research is based around that statement. Assume that either you are going to get hit by a bus tomorrow and your poor PI or your poor next grad student is going to have to come in and pick up where you left off, or that you're going to do all of this and you're going to publish a paper or try to publish a paper, and in 9 months when you send that paper out to peer review, somebody's going to write back and say this paper needs to have 5 more follow-on experiments performed. It's been 9 months since you touched this data. Make sure that after 9 months of not having touched this data, you don't have to rerun the same analysis that you already ran. You want to be able to run analysis on the new samples that you ran, but you don't want to have to redo anything that you've already done. So make sure that everything makes sense. Are you okay so far? I'm talking a lot, we're not talking too much. So, all right, do we have data? Has everybody been able to follow up on these and actually get a set of files made?
I'm not sure what that was.
So, did this run and come back to give you a prompt? Really is the only question.
Yeah.
Okay. Anybody who didn't see errors flying across the screen about no such file or directory, put up a red. Everybody else, we're going to move into the next thing. So, I'm going to give you my personal preference on the way that I deal with things with sequence data before we move into actually doing things. I like to have directories in my experimental set that tell me what I'm doing. So my raw data come into data. I don't ever touch it. Once it's in data, I never try to change those files. I try very hard to make them not writable, so that all I can do is read. I personally, and this is what you guys just got forced to do, make a QC directory where I do all of my trimming and my QA, QC analysis. And then I make directories for the steps that I'm going to do so that every data step goes into a new directory. You don't have to. You could just make an analysis directory and put all of your outputs into analysis. You could put everything in your home directory, but again, the more files you have in one place, the harder it is to figure out what you're doing. Unfortunately, the same is true for directories, so if you're doing a lot of different analysis steps, you might want to find other ways to do it. But this is sort of, again, the bare minimum suggestion of ways that you would want to do that. So now we have data. We have one set of samples that we're going to do something with. So I want everybody to go ahead and try to copy and paste that, and it should pop up a window. And while you do that, I'm going to catch up. So everybody, if they actually copied and pasted, should see something like this. Put up a red sticky. If you don't the bottom is just a repeat at the top for read 2. So what we're doing here is we're running fastqc to get a analysis of our read sets. So all of this percentage stuff is just saying I've gone through so much of the file. Because it's only 100,000 reads, it goes through it in about 10 seconds. The larger your read file, the longer fastqc will take to run. Again, this goes back to you have 2 options. You can either run fastqc first or not. The amount of time you're willing to wait for it to run may actually dictate that. So now what do we do with that? Well, let's look at it. So what happened? Don't do this. Okay. Now that I've cleaned out the directory and started over from scratch, this should be the same as what you all should see, and I'll move it up to the top of the screen so that you can see. So, when you run fastqc from the command line and you don't tell it to do anything, it makes some files. And in this case, right, our input files were these 2 fastqs, and it made an HTML file and a zip file. You can, if you want, download those HTML files and or those zip files and open it on your computer. Or you can do something like this. I'm going to shrink the screen so that the people in the back can see. Maybe. There we go. At least I will try to. Can't. How incredibly unhelpful of Windows 8. Okay. Again, we have these files. You can download them. However, the reason that I made you type all these fancy things and use the fancy mechanisms to connect to the HPC is that it enables you to use the HPC computing power to open up things like browsers. So, we can just look at it. Printing up a bunch of errors, but we're going to ignore all of the errors. And look, it opened up a new window with my output. So what is this? Has anybody used fastqc before? Hands in the air if you've used it, so I know whether or not I need to go through a lot of detail. I do. Okay. fastqc is a tool that does a couple of very basic things. It reads through your entire fastq file and parses to look for information about it. So the first thing it does is it gets the statistics. It says what's the name of the file, what kind of type file is it, which kind of fastq encoding was it. I could talk about that for hours, again, pet peeves. How many reads are there, how many were bad reads, how long were the reads, and what's the percentage GC. All that's just basic information, right? It doesn't make much difference, especially with RNA sequence, what this number is. If you're doing genomic sequencing and you have an organism with, say, 65% GC content, and this comes back at 40, there's a problem, right? That's something that either you sequence the wrong organism or something else. Sometimes you can expect, you could at least hope that the GC content for RNA-seq is about the same as the GC content of your target organism. I would lay very few wagers on that particular statement because expressed genes are not the same as the whole genome. So all of that's well and good, but usually you're never going to see any problems. What it does is it makes a summary on the right to say, these are all the tests that I ran. If it has a green checkbox, fastqc thinks that it's fine. it has an X or an exclamation point, it thinks it's really bad, or it thinks that it's questionable and maybe you should look at it. This is fairly standard. Oops, let me get just a little bit bigger. This is a fairly standard plot for FASTQ output. What this first per base sequence quality is exactly what it says it is. It took the quality of the read base by base. So it took all of the first bases in your read file and averaged the quality and said, well, the average is 33, and it's using box and whisker plots. Anybody here not know what a box and whisker plot is? I have to explain this to people, and it makes me crazy. Box and whisker plots are probably the most useful statistical plotting you can do. If anyone gives you a choice between making a bar chart and making a box and whisker plot, yell at them and explain to them why box and whisker plot is more important and that you should never give them the other choice. In this case, the box and whiskers are so small that it's just a flat line. And that's what you see all the way up to here, and even here you're starting to see a little bit of stretch. And you'll always see in a FASTQ file, as you get towards the end of the read, that you get more and more outliers towards the low quality. One of the reasons why you will hear lots and lots and lots of bioinformaticians telling you, I don't care what Illumina says, don't sequence 2 by 250, don't sequence 2 by 500, is because the quality starts to drop off and drop off and drop off. And when you get to the end of the read, all you've done is waste $500 or $600 per sample because all of the reads are useless, or even if they're not, they don't overlap anymore with the other half, and so the whole thing that you were trying to do was completely blown up. But what we're trying to do here is what we see is that sequence data starts to fall off, and we start to see some outliers go down to quality 20. Now, quality. Problems with quality? Anybody know what a quality score means? I don't. Really. I kind of know what it means, but it's also kind of a lie. Illumina calls quality as it calls its bases. So for every base, when it takes the picture of the sequencing lane, it says that spot has a base of this type at that point. And then it says, I can't be 100% sure, so how certain am I? And it puts up a number. That number is a logarithmic scale. So a quality of 2 is a 1 in 4 chance that it believes what it actually says. So it makes a base call and then says, it could be that, or it could be one of 3 other things, which inconveniently for you, the 3 other bases that you can get. So a quality of 2 all the way down here means that it had no idea and it just picked something. It goes up logarithmically, so by the time you get up to a quality of 40, which is the max that an Illumina machine will tell you it believes, is somewhere on the order of 10 to the minus 20 chance that it actually made a mistake on that base. So the higher the quality, the better. In this case, we're actually really happy with this, right? We don't have very many reads of poor quality down here. Everybody seems to be happy. We're still going to trim it. This is also a thing I disagree with, but it's kind of accepted de facto standard, but there isn't really any reason for it. When people used to do genome finishing, especially with Sanger sequencing, they would use 28 or 30 as the minimum quality for any base that they would incorporate into the genome. This yellow range was 20, which is half of 40, which is where a lot of people would also cut off. And then red is everything below that, which for a very long time they said just throw away everything under 20. Depending on what tool you're using and what analysis you're doing, you can use different cutoffs. I tend to err on the side of keeping as much data as possible and cutting off somewhere around 5 or 10. For most things I do. For RNA-seq, you want to err a little bit higher, so 10, 15 is good. Going above 20, at this point, in my opinion, is never a good idea at trimming quality. But again, don't take my word for it. Ask your friends who do this work as well, because they might have different experiences. Okay, so this is not useful for a small subset, but what it tries to do is show you in a read or in a lane whether or not there are tiles of that lane that might have had trouble. Again, you shouldn't have gotten sequence data back from the sequencing center where this looks weird. If it does, you should just go ahead and get on the phone immediately and check to see what's going on with it, and then move on with your life. And hopefully they'll resequence it if they have to. So we already did one set of analysis. We looked at the per base. Now we're looking at the per sequence. So every read, they averaged the quality along that read and told you what they thought what the average sequence quality for it is. Again, it looks fine. What they're really looking for is a semi-normal curve towards the top of the line. If you start to see bumps down here, be a little concerned. There was some DNA that went into the sample that it was really difficult for the sequencer to read. Why that is, I don't know, but you should think about it. The next one is the one that is based on assumptions that are not true for RNA-seq. So come on, give me back my prompt. I just lost my connection. This is one of the reasons why you might want to download your data instead, but we're back. So this is the one that I feel like is the most important to bring everything back to where we were. What are we looking at here? We're looking at, on a per base of your read sets, what the A, T, C, and G content are. Until you trim your data, you're always going to see junk at the front. That is usually adaptive sequences that haven't gotten trimmed out. However, there's another set of reasons why you're going to see, at the beginning of a lot of reads, you're going to see the same thing. Why is that? There's going to be some degree of stuff at the beginning that may show up as a present. On top of that, when you're doing genome sequencing, shotgun genome sequencing, you expect the A and the T line to overlap each other. You don't expect that from RNA-seq because you are no longer sequencing forward and reverse complements. You're only sequencing the actual forward bit. And on top of that, when you're dealing with eukaryotes, you always expect to see a little bit of enrichment of poly A for poly A tails, right? So it's going to look different, and it's always going to fail for FastQC. It's never going to like your per base GC content. Don't take that as an indictment of the actual data. Again, this is all built for whole genome sequencing. You expect a normal curve around the GC content of the organism. You're not going to see that with sequence data, so you can ignore that for FastQC. N content. This is actually not what you'll ever see. There will always be some N's in your reads, and you'll almost always see enrichment. What this means is that over the course of 101 base pair reads, you may see particular bases where you get an extra high peak of uncallable bases. It's an artifact in Illumina sequencing. It shows up. It will still be okay, but just be aware of it as you go through. Again, if you start to see the problem and it becomes concerning, call your sequencing center. Your length distribution. All of the reads were 101 bases long. Well, that's good because that's what you sequenced. We haven't trimmed it yet. It's going to look different once you trim. The last bits are the useful bits when you're talking about trimming and everything else. So there are sequence duplication levels. What it's doing is it is taking how many of the sequences fall into a category and then how many of those are duplicated when they show up, which is not really a useful stat for anyone other than a hardcore statistician. But the next two bits that come out of that are, so what sequences, when you see duplication occurring, are showing up very frequently? So it says, well, I found this thing a lot, 424 times out of the 100,000. It happens to be a TrueSeq adapter, which is good because as soon as we trim, it's going to go away. And then we found these two things that, funny enough, look a lot like the TrueSeq adapter, but aren't quite a perfect match, so I didn't know what to do with them. But it found those two, so it's concerned because of those extra sequences, which, again, when you see anything show up here, you don't necessarily have to be worried, but if any of them hit the TrueSeq adapter or any kind of adapter, you need to go back and retrim. And by the same token, at the end of reads, as this gets towards the last 40 bases, you start to see an upward creep of adapters showing up. When you do trimming, you want that to go away, right? If that's still there after you do trimming, then you trimmed with the wrong adapters. The last bits, again, are looking for k-mers that are overrepresented. Don't worry about it unless there's something really wrong with your data or if you're really interested in something later. So that was the quick and dirty version of what we're looking for in when we do FastQC. So we've done FastQC. We said, well, the data obviously needs to be trimmed because it has adapters in it. Sequence quality looks okay, but we're still going to trim. So we're going to do this, and I'm going to talk through what you go ahead and copy paste all of those bits in. Trimmomatic is a Java program. Java programs are incredibly difficult to understand what they mean. However, they're also easily portable. So you're going to run Java on Trimmomatic, looking for PE, which is paired end reads, because you have paired end reads. We're going to use 4 threads each so that we can get through it a little bit faster. And then it asks you, and I mentioned this before, why on earth you have to name every part of a read pair when both pairs are going to be the same name with R1 and R2? I don't know, but there it is. So you have to give it the names of the files. So here's your output file, or sorry, this is going to fail miserably. This may fail miserably. Let's try it and see whether it gives an error. I may have typed something a little bit too early. It should be input file, output files of the forward read, the unpaired of the forward read, the reverse read, the unpaired of the reverse read, and then a bunch of options. So I'm going to assume that when I copy and paste this is going to fail miserably. I'm going to double check right now. Yes. Okay, so if you copy and paste the way that I put it on the page, it's probably going to give you something that looks like this. Does that match anybody's experience? Good. That's not what it's supposed to do. I'm going to fix it on the page in a minute, but first I'm just going to fix it manually. Huh? Oh, you weren't talking to me. Okay, good.
Are we supposed to run the fast Q in 1.3 version first?
Sorry, what?
Never mind.
Oh, yes, because of the other bit. Sorry, I got too far. That's the problem. Ha-ha! I went too far. So we're going to do trimming in 2 pieces, and there's lots of reasons for this. But if we go back to the page, go away, have chat, you're not helpful. If we go back to the page, I just went a little bit too far. So we're going to do this first, which should work. And then once it's done, we're going to do the Trimmomatic. So this is not necessary. You can do all of the trimming in Trimmomatic. I just happen to like this tool because it gives you fancy charts and graphs and things. It does not, however, and this is the reason why I don't necessarily recommend that you guys use it, it does not, however, do any kind of good job at removing adapters. And given that there are adapters in almost every read set, you're going to have to do Trimmomatic anyway. So you can just use Trimmomatic from scratch without incorporating this step. I'm just showing you a couple of different trimming tools while we have them. So, once it gets through, you can actually see the report, which is the last thing. Events or Ghost View are Linux tools to open up PDFs. So in just a second, hopefully, I'll be able to show you what this report looks like, and it will look a lot similar with a couple of differences to FastQC. Yes, there's a typo. I apologize. So don't worry about it if it doesn't fail or doesn't pass, because we'll just look at it here. This is a really fancy version of that same plot. So this is base position, the box and whisker plot. This is the base number, and this is the quality. So what we're seeing now is a sort of 3D plot of the box and whisker instead. And all of the data is up at the very top, which is what you're looking for. Again, you can look and see a box and whisker of the reads. Again, you see it starts to fall down towards 30, but it doesn't drop any lower than that. Everything looks relatively happy. Lots more and lots more. I'm going to skip it because, again, I'm trying to make sure we get out of here by 3:30. So, once you have that, you will see that it has actually made the two files that we need for the copy and paste or Trimmomatic. So now the Trimmomatic command should work. So if we copy and paste all of those, it'll run really, really quickly and tell you what it did. So one of the reasons why I don't like Trimmomatic is that it doesn't do any real reporting. All it tells you when it finishes is that you came in with 96,000 reads, which means that the other trimming tool got rid of about 4,000 total reads. And of those, it only kept 88,000 paired reads, and then it dropped the rest. So, what happened that caused us to lose 10% of our data here? What do we do? Why would 10% of our data go away? Or at least 10% of our data become unpaired? I will give you a hint, though. All of these things at the end here are the things that it does. So the first thing it does is Illumina Clip, which is is a adapter removal tool. The next thing it does is filters at minimum length. So if a read gets shorter than 60, it throws it away. So Trimmomatic, because it's a Java program, you have to tell it what you want it to do. So PE is its paired end tool. So it's trying to treat both of the reads as pairs instead of just individual read pools. And then number of threads is just how fast you want it to run. You've got up to 20 cores usually to work with. We're just trying to make it go a little faster. And then file names and everything else. So when this runs, it gets rid of, what do we say, about 10% of its pairs? Right, so it ran, and both surviving. So what does this mean? This means that of the 96,000 read pairs, so there were 96,000 reads that survived after trimming, it got rid of another 8,000 of them, pairs, but it kept one of the two partners in most of those cases. So what does that mean? Does that make sense to anybody? No. Okay. I'll explain, but I expect you all to be more active. So what are we doing, right? We're taking two reads at a time and we're looking at them. The first thing that we did in the first tool was to look at the quality of those reads at the beginnings and ends and get rid of the chunks that were bad quality. We have an extra step here, which is the min length, which means that if we take this back and we start at 101 bases and cut and cut, and we cut all the way back to 59 bases long, so almost half of the read, it says I'm just going to throw this whole read away because I'm only allowed to keep reads that are longer than 60 bases. So I've got 2 reads. I've cut one back to 59 bases. What do? I can't keep both of those pairs. They're not pairs anymore. I have to throw this read away, and this partner gets to potentially stay in the pool, but not as a paired read anymore, right? So what I've done is I've created unpaired reads that sit over on the side and gotten rid of pairs. Make sense? So that's what we do with quality trimming. When we can do the same thing now with adapters. So what is it doing? It's looking at a pair of reads and trying to see whether either of those reads has adapter sequence in it and cuts off all of the adapter sequence. So what happens if the adapter sequence starts at base 42 of your read? It has to cut off 42 to 101 because there's no data past that point, right? So now you have one read in the pair that's 40, and another read in the pair that might be 60, might be 101, but it's probably about 40, well, might be 60, might be 101, but it's definitely not going to have a pair anymore. So it discards the bad read and puts the other read in an unpaired set of data. So what we've done in essence, well not in essence, but in practice, is gotten rid of another 10% of our reads because they're no longer paired. Some of that survives, right? So the forward-only read survives most of the time as opposed to the reverse read. It's a function of the way Illumina does its work that you read forward and then you read reverse, so you typically end up with much worse quality in the reverse read, so it always ends up being the poor sap who gets cut. So we only got rid of about 0.5% of our data in total, but we did have to cut a bunch of the read pairs. This is kind of important because usually most sequence or most analysis pipelines only use the paired reads. So in essence, even though you have the data for the forward here, unless you really want to get into the weeds, you're probably just going to ignore that too. So you're cutting down your data by 10%. 12% really when it comes down to it. Does that make sense? Are we okay? These are just things to be aware of. So the last thing to do in that tutorial is to just open up FastQC. So before we ran FastQC on the command line, however, when you connect to the HPC using an X connection, it can also open in interactive format, which means that you can just drag and use the file options. I'm going to open, and it starts in your root directory, so you want to go into the directory you're working in, which is RNA Seq model QA. And the outputs of that last set were trimmed and adapter removed, right? So we want, say we'll take the forward read, trim, adapter removed, and open it. And it's going to do the same thing that it did on the screen. It's going to print going through sequences and everything else, but now instead of it being a web page, it's sort of a semi-interactive thing. So per base sequence quality, again, looks okay. It trails off at the end, and as you go farther and farther down, it'll trail more. The sequence tile quality is a big blue square. We're not going to worry about it. The read quality scores don't really look that different. Again, we still see this because you're always going to see some function of that, so you don't worry about FastQC freaking out about it. GC content still looks choppy. But now, if we go down to the adapter content, we have a flat line. We've gotten rid of all of the adapter sequence. If you do your trimming and you don't have a flat line at the end, you've probably used the wrong adapter. Go back, figure out which adapter to use, and use the correct adapter when you're sequencing. That's all I can say about it. But now that we've done it, we've performed our task admirably. We have trimmed and chopped and adapter removed all of the bad stuff out of our read set, our 101 or 100,000 reads. we're actually ready to do RNA-seq work now, which has taken us an hour and a half, which is remarkably good, I think, for those of you that have never done it before. Once you do it again, it should only take 5 or 6 minutes.